West Nile virus is an arthropod-borne virus, or arbovirus, which are viruses that get transmitted through insects called vectors. The vector for West Nile virus is the mosquito. It gets transmitted in highest frequency through the female Culex species, which feeds on birds. The virus was first discovered in Uganda, west of the Nile, but has since been reported throughout the world. The virus causes a disease called West Nile fever, which normally causes mild symptoms, but can progress to full-blown encephalitis or meningitis. Normally, West Nile virus is found in birds and mosquitoes. Birds act as a reservoir for the virus, meaning the virus can replicate at high enough levels to cause significant viremia, or elevated viral blood counts, which allows for transmission to other uninfected mosquitoes. The virus will then replicate inside the mosquito and ultimately move into its salivary glands. So when the mosquito bites another animal, it injects its infected saliva into the host, since mosquitoes normally use their saliva as an anticoagulant. When the vector mosquito bites a larger animal, like a horse or a human, the virus can't spread from these larger animals because their blood doesn't reach high enough levels of the virus to be passed on to any mosquitoes that happen to bite them. And this is called a dead-end host. West Nile virus is composed of positive single-stranded RNA. This means that their RNA is actually mRNA, and the host cell ribosomes use this mRNA to make a long polyprotein chain, which is then broken into smaller pieces by viral proteases. This all happens in the cytoplasm of the host cell, since that's where ribosomes are found, and results in the production of several viral proteins. West Nile virus is surrounded by an icosahedral capsid, which is a spherical protein shell made up of 20 equilateral triangular faces. West Nile virus is also an enveloped virus, because the capsid is covered by a lipid membrane. Now, West Nile virus enters host cells through a lipid membrane protein called E2, in a process called clathrin-mediated endocytosis. Clathrin-mediated endocytosis is a cellular process that creates a vesicle to internalize a certain substance, in the case here, the virus, with the help of the proteins called clathrins. The virus's membrane will then fuse with the host cell's membrane, releasing its RNA genome into the cytoplasm of the host cell. The replication of the viruses then takes place, before exiting the host cell by outward budding of the host plasma membrane. After people are infected with West Nile virus, it takes 2 to 14 days to develop symptoms. Most people infected with the virus, about 80%, have few to no symptoms at all. About 20% of people develop moderate to severe symptoms. Moderate symptoms include fever, chills, headache, vomiting, or a rash. A very small amount of people, usually those with underlying medical problems, like the elderly or the young, develop severe symptoms like meningitis or encephalitis. These conditions can present with seizures, altered mental status, weakness, vision changes, severe headaches, and neck pain or stiffness. A unique feature of West Nile meningitis is the presence of a coarse tremor in the upper extremities. Laboratory findings associated with West Nile virus are nonspecific and include an elevated white blood cell count or elevated inflammatory markers like CRP and ESR. Cerebrospinal fluid analysis, or CSF analysis, typically shows pleocytosis, which means an increased number of white blood cells. CSF analysis can also show an elevated protein concentration because the inflammation leads to a more permeable blood-brain barrier, or BBB, which is a selective semi-permeable barrier between the circulating blood and the central nervous system. The diagnosis of West Nile virus is made by detecting about 4 times the normal amount of IgM in the serum or CSF, using an immunoassay test that's specific to the virus. A CT or MRI scan generates detailed, cross-sectional images of the brain that can help rule out other possible neurological diseases. In more severe cases, viral culture of tissues can also be helpful. Treatment for West Nile virus includes supportive care. If meningitis or encephalitis develops, steroids or other antiviral agents like ribavirin are considered, but only administered on a case-by-case basis. Prevention of West Nile virus remains the best therapy, using mosquito repellent and wearing long-sleeve shirts and pants. Alright, as a quick recap, West Nile virus is an arthropod-borne positive-sense RNA virus, or arbovirus, that gets transmitted through mosquitoes. The virus causes a clinical syndrome called West Nile fever, but can progress to meningitis or encephalitis in the elderly, those with significant health problems, immunocompromised, or young children. Diagnosis of West Nile fever is usually not clinically indicated unless findings of meningitis or encephalitis are present. When these findings present, a lumbar puncture should be performed for cerebrospinal fluid analysis to confirm the diagnosis. There is no specific therapy for West Nile virus, other than supportive care.